this from my kid's high school biology teacher, so credit to Miss Lenz, this is a DNA race. So my second favorite kind of science Jello. It's literally just the same process as making Jello. We add some hot water to this powder, we stir it up, we pour it into a mold, we let it sit, and then after 20 minutes or so, it's ready to be used. Okay, so what we're going to do is we're going to take our DNA and we are going to add it to these wells, and each well is going to get a different sample. So the lab that I do for my in-person classes is we do this mock crime scene. So the first sample is the DNA that we've extracted from the crime scene, and then each of the other samples are DNA that's been extracted from our suspects. Okay. And then we just and then we run our gel and we see if we can get a DNA fingerprint that matches from one of our suspects that matches the crime scene. Okay, but the way that this works is our agarose gel is this like gooey like substance that our DNA is going to diffuse through. And so if we kind of remember what are the things that affect the rate of diffusion, right? So if we have a really large molecule, it's going to be bonking in to stuff all the time, and it's going to move really slowly. But if we have a small molecule, it's going to be able to move really, really fast. Okay. So shape of our molecules also matter. So if we kind of roundy spherical things, those are going to move faster than long linear things. And in the case of gel electrophoresis, the charge is really important because we're going to put this gel and our samples, and then we're going to pour this buffer over it, and we're going to zap it with electricity. Okay, and so our negatively charged phosphate groups are going to be attracted to the positive end, and having that that the electrifiedness, it's not a word, I said it anyway, is going to increase the rate of diffusion. Okay, yeah. Okay, so let's look at how to interpret this gel photo, because if you look at this, you're going to be like, cool, Julia, don't know what that means, but I'll tell you what it means. This was my life for many, many years. Okay, so these are the wells. This is where we put the samples. This is the starting line of the DNA race. Okay, so all of our samples are going to, so our crime scene sample, suspect 1, suspect 2, blah, 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 so on and so forth. Okay, so each of these lanes is a different sample. In these, we're going to, you're right, on the two sides here, this is our ladder. This has DNA of mixture of DNA that are of known sizes. So we've actually taken a DNA molecule and we cut it up into various pieces, and that allows us to be able to compare the our unknowns, our samples, and compare them to what's happening here. So we can say, okay, the DNA in this fragment is probably about whatever this size is. Okay. All right, so there's two pieces of information that we can get from a gel photo. So the first thing is relative size. So we remember that big molecules are going to move slower and small molecules are going to move faster. So that means in that same amount of time, the smaller molecules are going to be further away from the starting line, further away from the wells. Okay, so they're going to appear lower down on the gel photo. So this fragment of DNA, we can conclude, is smaller than that fragment of DNA, and so forth. Okay, the other piece of information that we can get from these is the relative concentration. Okay, so the brighter the band, the more DNA there is of that size. So this one that's pretty faint, there might not be that much DNA of that size fragment, right? But this one that's pretty bright, that glows pretty good, we can say, okay, there's a higher concentration of that bit of DNA compared to that bit of DNA. We don't really know exactly how much it is, but just kind of like this compared to that, this one's more. All right. So we've extracted DNA, we did PCR, we ran gel electrophoresis. Cool. Now what do we do? And the answer is, yeah, it depends. Okay. So it depends on what the question is, what the application for the biotechnology is. I've done a number of things, including this RFLP, where we cut the DNA into all these little bits and then compared to see um, to determine if there was different species of fungi growing in leaves of plants. I've done Sanger sequencing. I've done this the newer version. It's called the next generation sequencing. I've done a million others that we won't go into the details of, but if you choose to change your path, right, and you want to learn more about these, at that point, that's when you can go and learn the details of those processes, because they can kind of be a lot. All right, but we will talk about one other mechanism, one other application of biotechnology.